Welcome to the introductory protocol video for the DNA barcoding lab. This is Chantal Lacroix, biology lab technician. We'll begin with the total DNA isolation. Please note that due to the time requirements of this procedure and the available lab time, steps 1 and 2 have been completed for you prior to class. Each group will start off with 2 samples of fish tissue, which were digested overnight in a 57-degree shaking incubator. Chantal will start by vortexing the samples. Next, she'll add 200 microliters of AL buffer and 200 microliters of ethanol to each sample. Then she'll vortex each sample. Using a pipette, she'll transfer each sample into a labeled spin column. Once both samples have been transferred, she'll centrifuge the columns at 8,000 RPM for 1 minute. She'll make sure that the centrifuge is balanced before starting, that is, that both tubes are opposite each other in the centrifuge. Once the spin is completed, Chantal will remove the columns from the centrifuge and place the column into a new collection tube. She'll discard the flow-through tube. Then she'll add 500 microliters of AEW1 buffer to each column. Then she'll centrifuge them again at 8,000 RPM for 1 minute. Once the spin is completed, she'll remove the spin column from the centrifuge and place the column into a new collection tube. As before, she'll discard the flow-through tube. Then she'll add 500 microliters of AEW2 buffer to each column. She'll then centrifuge the tubes at 14,000 RPM for 3 minutes. Once the centrifuge is complete, she'll remove the spin columns from the centrifuge tubes and place them into sterile microcentrifuge tubes. She'll then add 200 microliters of AE buffer to each sample. And the sample will incubate at 1 minute at room temperature. She'll then centrifuge them at 8,000 RPM for 1 minute. Once the centrifuge is complete, she'll remove the columns from the centrifuge, and this time she'll discard the spin column and save the microcentrifuge tube which contains the DNA. This DNA will be used in the next step of the lab. The second step in the DNA barcoding lab will be to set up PCR reactions for each DNA sample. Chantal will begin by labeling 2 PCR tubes with the sample names. To each tube, she'll add 12.5 microliters of the isolated total DNA. She'll then add 12.5 microliters of the primer cocktail to each tube. This is the tube that's labeled P. Finally, she'll add 25 microliters of the PCR master mix to each tube. This is the tube labeled MM. And finally, she'll seal the tubes with a cap. Once the samples are capped, she'll place them in the thermocycler and run the DNA barcoding program. When the PCR program is complete, the samples will be stored at 4 degrees Celsius, which we will then use for our Agarose gel electrophoresis. Third step in the DNA barcoding lab will be to run an Agarose electrophoresis gel of your PCR products. Chantal will prepare the samples by adding 2 microliters of 6X gel loading dye to 2 wells in a teriyaki tray. Next, she'll add 10 microliters of each sample to the loading dye. Of course, she'll need to adjust her pipette to do so. The purpose of adding the samples to the loading dye is, for one, to allow us to visualize the samples being loaded to the gel. Second, to make the sample more viscous so that it can load to the bottom of the well. And 3, to provide tracking dyes for the process of electrophoresis. This will allow us to monitor the progress of the gel as it runs. Now Chantal will load 3 lanes of the gel, one with 10 microliters of the DNA size standard from the tube marked S, and the other with 10 microliters of the respective PCR reactions mixed with the loading dye. Be sure to follow the gel loading guide in your lab handout. And if you make a mistake, just write the adjustments accordingly. After your samples have been loaded, run the gel at 100 volts until the dye markers have migrated 1/2 to 3/4 of the way down the gel. Remember, run to red. To make sure that the gel is running, look for bubbles cascading from the electrode. When our gel is finished running, we'll proceed with analyzing the gel. That part we'll cover later.